So the first question I was going to get to was, I would be from Sarah. She said, I would be interested in hearing a bit about antibody-dependent enhancement, both in terms of how it works and whether any of you have seen attempts to model it. And so the classic example of antibody-dependent enhancement comes from, and I'll explain what that is in a second. It comes from dengue virus. And so dengue virus isn't so different from some of the other viruses that we've been talking about, other than the fact that it is a vector transmitted virus. So it's transmitted by mosquitoes. It's in the same family as Zika. And it has the same structure where you have this membrane protein core around an envelope virus that then has its own surface glycoprotein, envelope glycoprotein. And trying to get my pointer. The envelope glycoprotein, coats the surface of the virus just like the HA or the spike. And it's a relatively complex protein that has different structural and receptor binding roles. And what distinguishes dengue biologically is that it has these 4 serotypes, meaning 4 distinct strains that are characterized by differences in how that envelope glycoprotein is detected by your antibodies. So when people talk about serotypes with viruses, what they mean is that they've defined antibody products from people or from animal models that can distinguish between those strains of virus. And before we had sequencing and all kinds of ways of phylogenetically characterizing viruses, the way that viruses were distinguished from each other was using actually antibody assays, where you would take serum or plasma from a recovered animal or human and look at reactivity of that plasma to a particular virus and see if it reacted or if it didn't. So there's 4 serotypes, and what distinguishes disease in dengue infections is that you get the first infection and it can be mild to moderate. It's rarely severe. You can then get bitten by another mosquito and get a second infection with a distinct serotype. So usually you cannot get the same serotype again. That's the way antibodies work. You will have neutralizing, sterilizing immunity to the same serotype getting it again. But if you get a different serotype, you can have a very, very severe response. So you can get a very, very severe infection, or you can get this incredible inflammatory response, which is referred to as dengue hemorrhagic fever, where you have this systemic shock-like syndrome, causes massive vascular leakage, bleeding from lots of different places in your body, and can be extraordinarily severe. But it only happens on the second infection. So this has been studied for a number of years, and theory has arisen around it and has been applied actually to other infections as well, including SARS-CoV-2, that what's actually causing this enhancement of the second infection is something that you've held over from the first infection. So the theory came about that it had to be actually something about your memory immune response that made that second infection so much worse. And so this developed into the theory of antibody-dependent enhancement. And I'm pulling this from this very nice review on dengue immunology that I've cited here. And so the idea is, in your primary infection, you mount the typical type 1 immune response that we've been talking about, which is you have dengue antigens that are presented by antigen-presenting cells, activate CD4 T cells, activate CD8 T cells, because it's a virus that's going to be inside the cell. And so we need CD8 T cells to kill them. And this, we're calling the serotype 1. And you make B cells specific for serotype 1, and they will secrete antibodies for serotype 1. And you will clear that infection and you'll be okay. And if you see serotype 1 again, you have these great antibodies and they're going to clear that infection and you're going to be okay. But now if you get a heterologous infection, meaning you're infected with serotype 2 here, there are 4 serotypes, so it could be any of those 4. Incidentally, and probably the reason why they drew it this way, the serotypes are just numbered 1 through 4, and there's a strong indication that getting serotype 2 second is actually worse for you than if you get serotype 2 first. And so here you've got your antibodies, but the way they're sort of representing this is you don't have very good antibodies. So here you have antibodies just totally coating the virus, and it can't get into the cells it wants to infect. Here you have this sort of marginal antibody response. They bind, but they don't really block the virus or pull it apart. And instead, what they do is they recruit the virus to cells that have receptors for antibodies. And there's a lot of cells that actually have receptors for antibodies in the professional immune system, because it's one of the ways that cells like macrophages and dendritic cells and all of these innate scavengers that we've talked about will go around and eat up debris. They'll look for an antibody and they'll say, well, whatever the antibody's bound to, I want to bind, I want to get rid of that. So I'm going to eat that up. And so they do that. The antibody binds to the dendritic cell and it's going to eat the virus. But the target cell for dengue virus is dendritic cells. So it's, again, another example of one of these viruses that actually directly attacks the immune response. And so like TB, like HIV, it's kind of clever and goes sort of after an Achilles heel of our immune system. So you have these non-neutralizing antibodies that can't fully block the activity of the virus, and the antibody actually does the work for the virus and saying, here's a dendritic cell, which is where you want it to go anyway. And so now the virus can get in the dendritic cell more efficiently than actually if you had never been infected with dengue virus before. And so that's the concept of antibody-dependent enhancement. It's not that it just doesn't work, it actually makes things worse for you. There are various flavors of theories around how primary infections can actually make your secondary infection worse. This is one where the immune system is actually actively facilitating the pathogen itself. You can also imagine situations like I just mentioned where the antibodies just don't work very well, which is just, as if you didn't have a primary response. And then there are ways in which the antibodies from the primary response can inhibit the development of your secondary response. And this is grouped under a topic called original antigenic sin, which is often discussed a lot in flu, but that's different from what's going on here. There, the immune response is inhibiting your immune response. Here, the immune response is facilitating the pathogen. So this could be shown in vitro very easily. It's a very robust phenotype. It could kind of be shown in vivo in mouse models, but this gets back to a recurring theme that we've been talking about in a lot of infections, including like malaria and TB, where the mouse models for dengue aren't great. And it really depended on the mouse model that you were using, whether you saw antibody-dependent enhancement. And so there was kind of some big debates in the field. There were people that were on the ADE side, and then there were people that were arguing that all of the bad things we see with hemorrhagic fever were actually coming from the CD8 T cells. And they had an animal model that they said could show that it was the CD8 T cells. And since neither animal model was really perfect and both had to be heavily manipulated, the field was a little bit in a debate, although for various reasons, the strength of the ADE argument kind of kept growing over the years. And so then Sarah's question was, has anybody modeled this? And is there any more, I guess, data about it? And the answer is yes. And there's this very, very nice paper that came out in 2017. It's not really dynamical modeling that they do in this paper. It's all different forms of statistical modeling, which I think Andreas has sort of introduced the concept of. But it was one of the really first, obviously correlational, but fairly definitive demonstrations that antibody-dependent enhancement is probably at play in the generation of the hemorrhagic fever phenotype. And so this is looking at a cohort, and the references here. It's looking at a wonderful cohort in Nicaragua that's run by Ava Harris and Aubrey Gordon, and it's been running since the sort of mid-2000s. It's one of the largest ongoing pediatric cohorts of any kind in the world. Initially established to look at dengue, now also looks at flu, RSV, SARS-CoV-2, and just an incredibly well documented cohort of children that have been followed for, with rolling waves of children that have been followed for almost 15 years. And so what they did is they had this, really precise longitudinal, at least annual, and usually much more frequently than annual sampling of kids. They knew exactly when they got dengue. They knew which dengue serotype they had, and then they knew what happened to them in terms of the phenotypes that they got. And so they binned kids. They actually did a number of different statistical analyses throughout the paper. And part of the sort of elegance of the paper, I think, is they show that regardless of whatever statistical regime you use, you get kind of the same answer. But this is in the first figure of the paper. They're binning kids in between, sort of prior to the infection, that they're going to look at whether or not a kid developed dengue hemorrhagic fever. So if you had never been infected before, or had very low memory, your antibody levels would be very low. And antibody levels here are being represented as a titer, which is like a ratio of a dilution at which point you can no longer detect the antibodies. So the higher the dilution it takes to dilute out the antibodies, the more antibody you have. This is a very common way that antibody is measured. And so these kids down here in yellow have the least antibody, and these kids up here in blue have the most antibody. And here they're doing a hazard ratio, and they're referencing to kids that had demonstrably never had a dengue infection. And so that's usually curated both on antibody levels and on medical history, where they looked very carefully of whether or not the kid had a dengue infection. And they could also use controls from seasons in which dengue wasn't circulating. And so what you can see is the hazard ratio sort of is relatively high, even at the very low levels of antibodies. And then it's even higher, and this is specifically for hemorrhagic fever or shock syndrome, at this 1 to 21 to 1 to 80, which is this sort of very modest level of antibodies that you'll see regardless of the infection. This is sort of the lower limit at which you would say you had protective antibodies to flu, for instance. 1 to 40 is sort of our cutoff in flu. And then it goes back down, and eventually as you get more and more antibodies, your hazard ratio starts trending to where it's in the negative region, which is what you would expect, right? If I have really good antibodies, I shouldn't get sick again, even though this is now with a distinct serotype. And that overall pattern actually tracks regardless of the outcome. So whether it's just severe dengue but not formal shock syndrome, and here you can clearly see the higher levels of antibodies are protective, whether you were hospitalized, and whether you had any dengue case. Now it starts to normalize. You see this protection, but one of the big discussion points in the paper is that you really needed to have really strict definitions of your outcomes and curation of your controls and your case subjects to actually see these effects well. And again, this cohort's just exquisitely suited for that. This is using a different statistical approach where instead of a hazard ratio, you're reporting an odds ratio. And again, you see the same thing where at the lower levels of antibodies, you have a much increased odds ratio for hemorrhagic fever and shock syndrome. And then even this 1 to 21 to 1 to 80 is really the critical window in which for all bad outcomes with dengue, you end up with that poor outcome. And here you're normalizing, or the comparison here, the odds ratio here is between a severe outcome and the non-severe outcome, non-severe dengue, uncomplicated dengue. And so that's all I had about that. I don't know, Sarah, if you had any other questions, or if anybody has anything they want to ask about that, this would be a good time for them. I'm going to switch to flies here in the next.
Yeah, can I ask a couple quick follow-ups?
Yeah, absolutely.
So first of all, on this slide and the one before it, are the children with higher antibody titers, did those also tend to be children who had several previous dengue infections, or is this controlled for that?
It's controlled for, it's controlled, my understanding is it's controlled for the number of infections. And I think actually it's set that they only had one documented infection, and it's actually adjusted for which serotypes they had as well too, so that it's not sort of biased in that way.
Okay. And then my other quick question was, with regard to original antigenic sin, is that something that also, would your second infection with a different strain also be more severe, like indirectly, just because your immune system isn't responding appropriately, or is that not necessarily the case?
That is the argument. It's not necessarily the case. So original antigenic stent has meant a lot of things over the years. And so it's one of these terms that kind of people like the term. And so they start using it, whether or not they're using it the way it was originally intended. But in the original formulation, it was a mouse model that actually it was described in. The animals with the prior infection actually did do worse and the argument was that it actually took them longer now to develop the right antibody response because they were being inhibited essentially in doing that by the previous antibody response, which was enough to sort of slow down the activation of the immune response, but not enough to slow down the virus. There's been more recent literature looking at people that get the flu vaccine every year, which is a lot of us in healthcare settings and in other situations, where for very specific strains of influenza, it looked like there was an original antigenic skin effect from getting the vaccine every year. And there wasn't so much that you did necessarily worse, it's just that it looked like you had no protection from the vaccine. And in that case, the arguments are about similar to sort of the dengue argument that they're making here, which is to trigger the response or to trigger the ineffectiveness, the two antigens that you're being faced with have to be sort of within a certain Goldilocks window of distance from each other. So not far enough away that your immune response knows that they're really different, and not too close that the antibodies work really well, but this sort of middle ground where the antibodies don't work well enough to really get rid of the virus, but they work well enough to slow your immune response down.
Okay, thank you.
The second question that I was going to address with just a few slides was from Sean, which is which principles, components of the immune system that I talked about are conserved in other types of hosts, especially insect vectors?
Well, there were a few more dengue-related one in the chat.
Okay, sorry, I didn't see. Let me pull up oh, there they are, yeah.
Also, I just put a link to a within-host dengue model. It addresses it not directly, but a little bit. And so the group that comes from Katja Köhler and her group, they've done a few dengue within-host models, the kind of models we're talking about here, instead of the statistical ones. So if anyone is interested in these kind of specific mechanistic models, look at this paper and whatever references they cite, and you'll get a nice sampling of those dengue models.
And I should mention this paper, the science paper, is the statistics were all done by Beth Talleyrand, who is our leader of our SISMID operation. So the first question is, do these mechanisms match up with issues with the vaccine? That is a great question. And so when people propose the idea of doing a dengue vaccine, a lot of immunologists were like, that is the craziest idea in the world in a way, because the whole point of almost all the severe disease in dengue is a secondary infection. And now you're proposing a situation where you're going to give everybody the equivalent of their primary infection, which now just like creates this giant susceptible pool. And so there have been a few different versions of the dengue vaccine proposed and gone into trials. And one of those trials did have to end early and was a gigantic disaster, because that's exactly what happened. People in the trial were actually more likely to get severe dengue than if you were in the placebo group. There are other formulations of the vaccine where they're trying to give you good antibodies to all 4 serotypes, and some versions of those are still in trials. And so, yeah, there's a lot of, there's a great literature actually on that failed trial, and a lot of finger pointing now that it kind of met expectations of a lot of people.
And isn't it right now that the dengue vaccine is only recommended for people that have already shown to have had at least one dengue natural infection for kids?
I think that was one of the ways they were, addressing the study designs going forward. And there were 2 major, I haven't gone back and looked at that literature recently, but there was one that was like a real failure. And then there's another one that I thought was still kind of moving forward.
Yeah, but didn't they have a major rollout in, was it Philippines or Indonesia? And then it went wrong and they had to stop the whole thing? Yeah.
There's like a big disaster story around one of them. Sean asked the question, what has led people to suggest that ADE might be a factor with SARS-CoV-2? And so that comes from research that was done shortly after SARS-1, also in vitro models, and also with SARS-1, we don't have great animal models. Also with SARS-2, we don't have great animal models. And so you could show this antibody-dependent enhancement in like a half a dozen papers in SARS-1. And so then when SARS-2 hit, And you had this weird effect of most people do really well, but then some people do really bad, and even some young people do really bad. Was there some interaction with prior infections, particularly like prior coronas? You know, if you were following the SARS literature early on, SARS-2 literature, it was just like a field day of commentary and speculation. Not unwarranted, but lots of tweets about ADE, some very, very prominent immunologists. I think, you know, maybe because of the dengue stuff that just happened with that vaccine about ADE. And now there's a general sense that this is really not probably driving any of the pathology that we see, at least so far. And it's kind of fallen off as a major point of concern. And it just goes to show sort of, you can get a phenotype in vitro, and it doesn't mean that it's anything in vivo. And then, has the antibody avidity been explored for this effect, or just titers? Definitely has been explored in a lot of the in vitro models and in some of the animal models. There's a huge literature on ADE and dengue that goes back, you know, 20 or 30 years at this point now. And so that avidity, and that's, again, it's this Goldilocks, like avid enough to bind, avid enough to get you to the dendritic cell, but not avid enough to actually bind enough to block the functions of your envelope protein over the whole virion. One really interesting piece of data that is out now is that part of why Zika was bad was probably because, in certain areas, was probably because of an ADE effect, and that Zika and dengue, which are both flaviviruses, could serve as the first hit for each other. So if you had a dengue, your Zika could be worse. If you had Zika, your dengue could get worse. And so there was enough cross-reactivity between them to create the ADE effect.